Bismillahirrahmanirrahim. Dear students, hope you all are well and doing great. The video on this topic contains instructive and educational content that is both shareable and easy to retain. So let's start. My presentation topic is about descriptive embryology that describes the developmental stages of an embryo. It includes all the developmental stages of an embryo from a fertilized egg to the formation of all the major organs of an organism. So here is the outline of my topic. On this video, we will discuss about fertilization, embryonic development, cleavages and egg types. Then we will see the primary germ layers and their derivatives. Then we see Echinoderm embryology, and the last is vertebrate embryology, in which we say Chordate body plane, Amphibian embryology and Avian embryology. So first of all, fertilization. Fertilization is the fusion of sperm cell nucleus with the egg cell nucleus to produce a zygote. The stages of fertilization can be divided into four processes. Number 1 is sperm preparation, number 2 is sperm egg recognition and binding, the third is sperm egg fusion, and the fourth one is the fusion of sperm egg pronuclei and the activation of the zygote. For the fertilization to occur, a sperm must penetrate the gel coat of an egg, which consists of protein. To penetrate this coating, the sperm of most animals possess enzyme called sperm lysine present at the head region of the sperm. In a simple word, fertilization is the story of all of us. During sexual course, about 300 million of sperm enter the vagina. Sperm pass the opening called cervix and enter the uterus. Then sperm make contact with egg. How can sperm make contact with egg? Now we will discuss all the four stages of fertilization. Take a look on the picture. The sperm make contact with the egg. The first event is capacitation. As you know, at the head of the sperm, different type of protein are present. The sperm has to get rid of this kind of protein. So cleaning of sperm head is called capacitation. Capacitation increase the motility of the sperm. Now next thing happening is the sperm movement or migration towards zona pellucida. At zona pellucida region have specific zona pellucida protein called ZP proteins. The sperm also have specific protein that actually recognize the ZP protein. So sperm interact with zona pellucida. Sperm head have special organelle at the head region called acrosome. Once sperm attach with zona pellucida, acrosome release enzyme that start boring a hole through zona pellucida. This is called acrosomal reaction. After that, sperm face another barrier called cell membrane. Cell membrane has specific protein called alpha and beta protein. Once sperm bind with the membrane, special channel open and sperm push in. Once the sperm cell push in, it triggers the specific action in the egg. The smooth endoplasmic reticulum of the egg produces calcium ion. This calcium ion starts to migrate toward cortical cell membrane, and lysosomes also get fused with the membrane and release enzyme. And they start to degrade zona pellucida and harden the cell membrane. This prevents polyspermy. Now egg and sperm nuclei fuse to form a zygote. Now egg activation. The fusion of acrosomal and egg membrane is the beginning of egg activation. Basically, it is a series of biochemical changes in the egg that ensure the completion of the fertilization and initiation of the embryonic development. In this picture, you can see egg activation. Calcium ion is producing that block the polyspermy. Now membrane and cortical events. Some of the earliest changes in the zygote occur at the plasma membrane in the outer region of the cell cytoplasm. These early changes ensure fertilization by only a single sperm. As I told you, there is 300 million sperm that enter the uterus. So these changes allow only a single sperm to make contact with egg. Now embryonic development, in which we see cleavages and the egg type. Cleavage refers to the cell division that occur during embryonic development, and the cell that cleavage produce are called blastomeres. Egg size, cleavage pattern, and the length of embryonic period of animal species are related to the quantity and the distribution of yolk in an egg. There is a two type of cleavage pattern, holoblastic cleavage and the meroblastic cleavage. Cleavage that divide an egg is Cleavage that completely divide an egg is called holoblastic cleavage, and if cleavage cannot completely divide the embryo because of large quantity of yolk, then the cleavage is meroblastic. Now the type of egg according to the yolk. Number 1 is alethal egg. In alethal egg, yolk is very little or none. The type of cleavage in this type of egg is holoblastic cleavage. For example, in mammals. The second is centrolithial egg. In centrolithial egg, yolk is present at the center, and the type of cleavage is superficial cleavage. For example, in insects. The third is isolithial egg, in which yolk is evenly distributed and the cleavage pattern is holoblastic. As a result of holoblastic cleavage, blastula are formed. For example, sea urchin, sand dollars, etc. The third Next is mesolithial. In mesolithial egg, yolk is unevenly distributed and cleavage pattern is holoblastic. For example, amphibian egg. The last one is telolithial, in which very unevenly distributed yolk is present and the type of cleavage is meroblastic cleavage. For example, bird, fishes and reptiles. In the second picture, you can see holoblastic cleavage and meroblastic cleavage. In holoblastic cleavage, this cleavage divides the embryo into two equal halves. But in meroblastic cleavage, due to presence of large quantity of yolk, embryo do not divide into two equal halves. The next topic is primary germ layers and their derivatives. Tissue organ of the animal arise from layers, and these layers are called primary germ layers. There is three type of layer: ectoderm, endoderm and mesoderm. One layer, ectoderm, give rise to the outer body wall. The endoderm form the inner lining of digestive cavity, and mesoderm give rise to the tissue between ectoderm and endoderm. Some undifferentiated mesoderm develop into muscles, blood vessels, skeletal element, and other connective tissues. Here you can see the three germ layers and their derivatives. First is outer layer, ectoderm, that give rise to the outer surface, central nervous system and neural crust cells. Then the middle layer is mesoderm, that form notochord, bone tissue, tubule cells of kidney. It form blood cells and facial muscle cells. Then the innermost layer, that is called endoderm. Endoderm give rise to digestive tube, pharynx, thyroid cells, respiratory tubes and lung cell, etc. That is all about germ layers and their derivatives. Now echinoderm embryology. The egg of echinoderm have relatively little yolk or evenly distributed. Cleavage are holoblastic and result in solid ball of mass called medulla. Afterward, a fluid-filled cavity, the blastocele, is formed. The embryo now a hollow sphere called blastula. The next step is gastrulation, in which invagination of the cell at a point in the vegetal half of embryo take place. The point of invagination in the blastopore, which will eventually form the anal opening of the larva. During invagination and embryonic gut, the archon tone elongate and reduce the size of blastocyst. The embryo begin to lengthen and assume a pyramidal shape, as you can see in the picture. The next is vertebrate embryology, in which first of all we see chordate body plan. There is some general characteristic of chordate. The chordate nervous system developed from ectoderm and is dorsal and tubular. The nervous tissue proliferates into brain. The notochord is the primary axial structure in all the chordate embryo. Notochord is mesodermal in origin and it is flexible, sportive, and lies just beneath the neural tube. In addition, all chordate possess pharyngeal slits and post-anal tail. As you can see in the picture, the dorsal neural tube notochord is present just beneath the dorsal nerve cord. There is also present pharyngeal slits and post-anal tail. Now, amphibian embryology. The general characteristics of amphibian are the following. Most amphibians lay egg in watery environment. Amphibian egg usually develop with little care or protection from the parent. Frog egg have pigmented animal pore. This pigmentation help camouflage the developing embryo from the predator. Now, initial cleavages and gastrulation in amphibian. The first cleavage of the amphibian embryo, like that of Echinoderm, is longitudinal. It begins at the animal port and divides the grey crescent in half. Because of presence of large amount of yolk, cleavage is slower at vegetal pore. Therefore, amphibian morula consists of many small cells at the animal pore and fewer large cells at the vegetal pore. The blastula form in much the same way as in the Echinoderm. Then gastrulation in amphibian. The first type of gastrulation is involation, in which inward movement of cell take place. The blastopore extend and the end of the blastopore join, forming a yolk plug, and the blastopore eventually closes. The coda mesoderm begin form the notochord. The embryo lengthen during gastrulation, and after gastrulation nerve cord formation begin. The mesoderm proliferate to form somites and body cavity. You can see gastrulation in a frog embryo. The inward movement of the cell take place and blastopore is formed. Then the blastopore ends join and form a yolk plug, as you can see in the picture. Afterward, coda mesoderm begin to form the notochord. Embryo lengthen itself and a cavity or contorn cavity is developed. That is all about gastrulation in frog embryo. Now, avian embryology. Chicken egg used model for the development of bird and reptiles. The egg is released into ovid egg where fertilization occurs. Following the fertilization, membrane and fluid collect around the egg. The white part of the egg consists of water and protein called albumin. This watery environment protects the egg from mechanical damage. In addition to denser strand of albumin attached to the inside of the shell and to the egg, and suspend the egg in the center of watery albumin. The shell is made up of calcium carbonate impregnated with protein. Thousands of tiny spores in the shell permit gas exchange between the embryo and the outside. As you can see in the picture, at the center is yolk is present and a albumin or white portion surround the embryo and protect it from any mechanical damage. Now, early cleavages and gastrulation in avian embryo. Cleavage of the chick egg is meroblastic, means it do not divide the embryo into two equal halves due to presence of large quantity of yolk. As a result of this cleavage, blastoderm is formed. Proliferation and movement of the blastoderm cells sort the cell into two layers, epiblast and hypoblast. The movement of blastoderm is the beginning of gastrulation. A medial linear imagination called primitive streak gradually extends anteriorly. A depression called Hansen nerve form at the anterior margin of the primitive streak and mark the beginning of an inward migration of epiblast cells. Migrating cell from mesoderm. Epiblast cell on the surface of the embryo is the ectoderm, and the hypoblast form the endodermal lining of the gut tract. And the three germ layers are now arranged above the surface of the yolk. As you can see in the picture, epiblast cell, hypoblast cells, and blastocele, a cavity developed due to migration of the cell. The last topic is the development of extraembryonic membrane. The chick embryo possesses three extraembryonic membranes called amnion, chorion, and allantois. The amnion encloses the embryo in a fluid-filled sac. This amniotic cavity protects against shock and drying. The chorion is near the shell and becomes highly vascular and aid in gas exchange. And the removal of nitrogenous waste takes place through allantois. As you can see in the picture, three extra embryonic membranes develop around the chick embryo. These layers protect the embryo from any mechanical damage. That is all about my presentation. Thank you so much. Allah Hafiz.